but it is my great pleasure to introduce Dr. Alice Telesnitsky to give the first keynote of this year. I think it's fitting that she follows the RNA session because she's worked a lot on retroviral RNA. Just a few words about Alice. I'm sure she'll tell us more about herself, but she is a fellow avid gardener, as you can tell behind her. She's also a fellow West Coaster. She got her bachelor's degree at UC Davis in genetics. And when she graduated, she worked for a couple of years at a relatively new biotech company in California. You may have heard of it, Genentech. And she even got a book chapter. I think she was a co-author on a book chapter while she was there. Then she did her PhD in biochemistry in Michael Chamberlain's lab at UC Berkeley. And she traveled all the way to the East Coast to do her postdoctoral fellowship at Columbia in Steve Goff's lab. And she had an American Cancer Society fellowship and an NRSA fellowship, which was great. When she left Steve's lab, she became an assistant professor at the University of Michigan in the Department of Microbiology and Immunology. And she's been there ever since. She's now, of course, a full professor. And it's notable that she is the lead PI on one of the 5 NIH U54 HIV structural biology program grants, the Center for HIV RNA Studies, or CRNA. And you've probably, if you've been to Cold Spring Harbor, you've probably met or seen Alice. She's been very active in attending. She also coordinates the Andy Kaplan Award every year, which is great. And one thing, if you've looked at her CV or you just know her, you know that she's very active in teaching and mentoring. Not surprisingly, to me at least, she's won several teaching and mentorship awards. And she's very involved in science education and advocacy. And I just wanted to say that I remember the first time I met you, Alice, it was at Cold Spring Harbor. I had given my very first Cold Spring Harbor talk, which was nerve-wracking, but you came up to me, you found me during a break, and you told me I did a great job, and you asked me some questions, and it really meant a lot to me. It was very sweet of you, and I still remember that, and so I consider you a mentor and a friend, and I look forward to hearing what you have to say, probably about retroviral RNA in some form. So please take it away.
Well, thank you very much. And I guess I'll go ahead and share slides. And it would be very nice for me to talk a lot about retroviral RNA, but I think I somewhat foolishly made this decision that I was going to go for more on the sort of reminiscent side, which we sometimes have had people do. So I was going to talk a little bit about my career and introduce myself to people who don't know me. So I guess I've been coming to Cold Spring Harbor for over 30 years now. So I qualify as one of the old people. And I guess I just figured I don't get to really expose myself in this way all the time. So I was going to tell you more about my influences in my life. And I'll just sprinkle it a little bit with really some of the old science that we did with mechanistic aspects of reverse transcription. So I start off with my life. I mean, I was a native Californian, as Andrea pointed out, K through PhD in the California public schools. There I am. Probably my parents were the biggest influence. Isn't that for every kid? My mother was really intimidatingly brilliant, probably the smartest person I ever met in my life. She worked at UCLA when I was a kid in the Chancellor's Communications Office, which in those days was composed of a team of really hard-hitting, overeducated women who processed all the information that flowed into the university and distilled it into policy proposals. And then they gave it to the chancellor and other talking heads who usually enacted it without changes. So my father, on the other hand, was the artistic one in the family and probably responsible for conveying in me some of my inappropriate sense of humor. He was a lab tech, and he gave us children styrofoam packing boxes to build forts out of, and he made weird but wonderful hydroponic plantings in chemical glassware with tropical fish swimming around the roots. So our family was one where life was an adventure and creativity was always valued over practicality. But despite all this, I sort of always knew I was going to be a scientist. And so that was why it was sort of disorienting for me at the end of my college career, when for a couple of years I thought I wasn't going to be a scientist after all. But then one day I met Herb Heinecker in an elevator. And early in 1981, we had, as Andrea already pointed out, just had this spectacular initial IPO of Genentech, and they were looking to expand. And since as an undergrad, I had worked in molecular biology, I knew a little of the lingo and a few of the techniques, then I could join this ragtag team of people who were vaguely qualified to work in biotech, the tofu makers, the psilocybin farmers, and the others, and go and make great discoveries in molecular biology. Meanwhile, Herb himself sort of devoted most of his energy to a spin-off company. It was called Genencore, and one of his first main things there was to clone industrial enzymes, including the enzymes that are required to convert cornstarch into high fructose corn syrup. So cloning these enzymes transformed the ability to make high fructose corn syrup and made the process much less expensive. So when you think about what influence molecular biology has had on public health, you have to admit that this has been a really huge contribution that Herb made. Okay. Meanwhile, I was working for another Dutchman, whatever you call those guys, named Herman de Boer, and his claim to fame was making hybrid promoters in E. coli. That, for example, the TAC promoter had the strength of the tryptophan promoter and the inducibility of the lac operon. And so while I wasn't just actively working on expression constructs that came in later, helpful when I was making expression constructs for reverse transcriptase, then we did company-wide initiatives, like everyone would be given a stack of bacterial plates and a bunch of toothpicks, and we all had to pick out the plaques one by one into microtiter plates for these big cloning blitzes that we would have. And I think that I have never had such a wild couple of years as working at Genentech. You know, the whole play hard, work hard ethos, we could talk about that for forever, and people jockeying for space in the speed back at 3 a.m. But eventually I decided to go back to school, and I went to Berkeley. And at Berkeley, then I worked in the lab of Mike Chamberlain, who was studying transcription. And my first paper in that lab was published back to back then with a paper from the Goliger lab at Cornell. And what we found then was an unusual or sort of unanticipated linkage between the initiation, elongation, and termination phases of the transcription cycle. So specifically what we saw is sequences in the initial transcribed region would affect whether or not a terminator far downstream was used. So we still don't know how this works, but some people have suggested that RNA polymerase, which I'm showing you with a brown oval here, when the nascent RNA in red here was coming out of the exit channel of the RNA polymerase, perhaps that 5' end sequence would then, either through transient or more stable interactions, affect the architecture of the exit channel in a way that would then allow it to recognize terminators far downstream in a different way. Other people have said, well, maybe just this, somehow this RNA could affect the actual structure of the terminators, even though it didn't matter what the terminator's context was or whatnot. So in order to suggest that, then we have to suggest that somehow that 5' end would stay with the elongating polymerase and affect its activities in ways other than this proposed architectural role. But at any rate, in potential foreshadowing of other things we're doing now, we saw that RNA 5' ends affected the transcript subsequent phase. So Mike Chamberlain lived up to his reputation of being a very tough mentor. We had to do things, periodic things that the trainees had to do is we had to make our own alpha labeled nucleotides. We would start with 100 millicuries of P32 and dive into anhydrous chemistry for 2-day procedures. And he was very rude to us and yelled at us all the time. But we also had a lot of fun. So for example, probably our lab blender was used to make margaritas almost as often as we used it to grind up human placentas to make RNA polymerase preps. But towards the end of graduate school, I started thinking, I want to work on polymerase mechanisms. I like this enzymology, but I don't want to stay in E. coli. I want to work on another polymerase. And I really wouldn't mind working for a more normal sort of person. So one evening I wandered down to the TEAJ lab on the second floor of the biochemistry building, got to chatting with the postdocs there. And this woman then convinced me somehow that reverse transcriptase was definitely the up and coming polymerase for me, and that Steve Goff, she thought was a sort of normal person. So I should go and follow up on her graduate work. So that's how I ended up in the Goff lab. And it really was a very wonderful time and working in the Goff lab. And I was there for 5 years. And along the line, I got to write some chapters for some books about reverse transcriptase. So not only was I doing mechanistic reverse transcriptase studies and learning virology by doing some complementation between RT and integrase, but I got to work and write this chapter in a book that was edited by that pillar of retrovirology, Ann Skalka. And then later on, I helped with a chapter that was edited by Coffin, Hughes, and Varmus. And fangirl that I am, I have held on to some of those edits all these years later. So one of the things I really appreciate about Steve is he created a lab where I and previous and following on trainees have felt comfortable to have children. But I think other aspects of my life in science as a woman didn't always go so smoothly. So for example, being obviously pregnant with my second child when I was interviewing for faculty positions was probably a tactical mistake, because it seemed that everyone wanted a woman, but nobody thought that a mother was up to the task. So being keyed into that thought, when the University of Michigan called me at the end of the interview season, then I got some very quick thinking that made me say to them, oh, well, I can't come out right away like you want me to. It's going to be at least, and I pushed it as far as I could, 3 weeks. Okay, 3 weeks. So they said, well, okay. And then fortunately, at my next prenatal visit, as I said, I was already more than a week overdue, the doctor said, well, why don't we induce labor? And I said, well, I told her, okay, let's do it, because I knew with that timing, then my child's birthdays in the future years would always be before Cold Spring Harbor. So then we had Ben, and then some of you old timers will remember Ben at 6 days old giving his first poster at Cold Spring Harbor. So then the next week I went out to Michigan and I was lactating up a storm, which meant I had to keep running to the restroom after every office visit, but otherwise it went fine. And so then they hired me, they gave me a job, and I went to Michigan. And having done graduate work at Berkeley and then lived in New York City, adjusting to Ann Arbor, Michigan was a bit tough at first, okay? So at first I thought my faculty colleagues were joking when I found them sitting around discussing sports stats. But no, there were tailgates and golf outings and times when March Madness was pretty much all that anyone would ever talk about. Meanwhile, some well-intentioned faculty colleagues warned me about seeming unprofessional when I helped a graduate student organize a holiday cookie exchange. So to put that in context, this is what our faculty looked like half a dozen years before I joined. Then my colleague Ovita Fuller joined. She became the first woman tenured in my department's 100-year history shortly after I joined. So today, over half of our faculty is women, and we still have our holiday cookie exchange. With my first graduate student, we dissected steps in the process of reverse transcription, but eventually we came to think more and more about genetic recombination, especially when one Fun joined the lab because he wanted to work on HIV. I had been long interested in the possible functional differences between HIV and MLV-RTs. When I had been in Steve's lab, Millie Georgiadis had solved the crystal structure of the core of MLV-RT using enzyme I had expressed. And not long after that, Laurie Kolstad had solved, or long before that, Laurie Kolstad had solved the structure of HIV-RT, okay? And so also around the time I started my lab, or a little bit before, Howard Temin and Wei-Su Hu's studies with the gamma retrovirus, spleen necrosis virus, had shown that about 1 in every 7 viruses was recombinant. But the early work from Doherty's lab and the highly mosaic nature of HIV genomes suggested that recombination for that virus might be even more common. So was recombination our potential functional difference to study that we could look at structural determinants of? So we couldn't really tell from the literature because people had used different systems. So we wanted to set up our own 2 vector recombination assays, similar to what other people had, but using the same target systems for both HIV and RT. So we're looking here for the co-segregation of this pure resistant marker and this LAC-Z, which had been on separate RNAs. So co-segregation then would be an indication of recombination. And we found out with these assays that lo and behold, the HIV recombination was 5 to 10-fold higher than that for MLV. But then before we got into the mechanistic biochemistry, we wanted to develop 1 assay vectors because it was kind of tedious to have to get the 2 into one cell, or into one virion. So one of the ways we did this was to make a vector that was based on oncogene transduction models. So in those models, the retrovirus inserts in front of the oncogene, a read-through RNA is made, that RNA is packaged, and then during subsequent infection, 2 non-homologous template switches can patch the oncogene between the 2 LTRs. So we made experimental vectors that were sort of like that, putting our donor of sequences up downstream of a deleted polyadenylation signal. So every virion would have donor and acceptor for recombination studies, and we would then see our co-segregation of markers in this way. And in this case, then there was no difference between HIV and MLV. So we were left with a situation where HIV recombination was much higher when measured by marker segregation using 2 RNAs, but indistinguishable when they were on a single RNA. So that suggested to us that this was not an enzymological RT phenomenon, but instead had something to do with template availability. And whereas we and everyone in the field had assumed that RNAs would come together at random to assemble and that half the particles, if they were equally expressed, would be heterodimeric and the other half homodimeric, what would happen if there was preferential self-association? We might still have a few heterodimers, but only most of them would be homodimeric. So a student in the lab, whoa, a little out of focus, went in to study this, and she did a pull down assay, basically pulling on one of the RNAs and seeing which dimers would come down and what RNAs would be free. And when she looked at this, lo and behold, what she found there was that HIV RNAs did dimerize in the proportions that would be predicted by random association. But in the case of the MLV RNAs, they were clearly self-associating for dimerization, and the magnitude of this fully explained the recombination differences. So we had this model, and what we thought was happening then, and you remember the 2 pools of MLV RNA from Judy Levin and so forth, we figured that the RNAs were either dimerizing while they were being transcribed, or else some RNAs were not dimerizing. And if they were staying monomeric, we assumed that they would export and join an mRNA pool. And somehow these ones that were dimerizing in the nucleus would have to move to assembly sites without succumbing to the cytoplasmic ribosomes and things like that, because they were still in the same partnerships that we had seen. So this was only a model, and we had to do more work. But unfortunately, at this stage, we got scooped. So there was this group in Belarus, of all places, that went ahead and had this evidence for co-transcriptional dimerization before we did. But then we published, and then a group in Denmark published. And in many ways, it's good to have competitors. It's tough to be scooped, but it's nice to have people who are interested in the same kind of stuff you are. And if you're not sometimes totally in awe of your competitors, then really, you must just not be paying attention. So one time at Cold Spring Harbor, there was this fellow named Thomas Rummel, and he was presenting a poster that had pictures of Prague, and he was inviting us to a retrovirus assembly meeting. And I thought, that looks cool. And I'm interested in how RNAs get into virions and that sort of assembly, so I could go. So I went twice, and it was fun, except for the time that Eric Fried and I got Giardia. It was really quite fun. And then one afternoon, they took us to a brewery, and I guess I got kind of a little loosened up by a Pilsner and started talking kind of crazy in the ways I do about things that I was interested in, you know, like the most cool thing in the world being 7SL RNA, that host RNA that's packaged into retroviruses in proportion to GAG, independent of genome. So you could measure how well the genome was packaging by looking at the ratio of genome to 7SL. And I don't really remember this too clearly. But then 3 years later, out of the blue, I got this call from Mike Summers, who had been at that meeting, who actually presented very interesting work, asking if we would run some packaging assays for him. And I'd never really planned to work on packaging per se, but my genetic recombination grant had just been triaged, my competing renewal. And he offered to help us out. So we jumped into this collaboration, and it was the most unexpected thing, but in the long run, it's been one of the most meaningful. Mike, if you're listening, it's been a wonderful thing that's ongoing now for 15 years or so. Unfortunately, when Mike was renewing his R01 and we were going to actually get a little bit more money, I got an application for an overqualified postdoc. And it was Sergei Hartanchik, the fellow from Belarus who had scooped us in the MLV dimerization. But fortunately, he joined our lab in 2010, and he's been with us ever since. And I'd really like to tell you more about the RNA studies, but I don't have a lot of minutes here, so I'm going to cheat and tell you, register for this fabulous structural biology meeting coming up in just a month. It's free. Mike will be talking a lot about our collaboration, and it is really exciting. Victoria and Jinwei also have super, super cool stories, so I encourage you to listen in. But going back to the past, I wanted to get back to that enzymology that I had told you about. And we always did want to study this template switching from defined positions. And the story getting there is interesting too, but I'll just jump straight ahead that those women who I just showed you set up a system to study non-homologous recombination. And basically what we did, because we wanted to be able to have recombination go to various sort of random places, is we did this within an artificial intron. And we could design this so from the point that we thought we were going to have transfer to where we thought we were going to have the acceptor template, we only had maximum of 2 nucleotides. And the question was, what is the virus going to do when we have this strong pressure to make an intact puro with a splice in the middle? And what did it do? We thought it was going to use those microhomologies, but no, vastly over 65% of the time. And because of the high selection, we could sequence over 100 of these guys. What we saw was what I call splinted recombination. So it was really rare, weird things. And of course, the titer was low. But what we saw then was that occasionally the polyadenylation signal on these vectors also had been read through, and there would be downstream sequences. And sometimes then the transferring polymerase would fortuitously find more homology in these little patches than we had intended it to find. And then it would continue until it had fortuitously found more homology again than we had intended to find. And so we saw, as I said, dozens and dozens of these things. This was basically what RT does. So what non-homologous recombination happens is very often then achieved by a short patch insertion where there is the donor and the acceptor, but in between there's this third sequence. And that third sequence is a physically separated sequence from either the virus or from the host. And we were certainly not the first people to see this sort of thing, but I think that us quantifying this pointed out how this is huge. This is what viruses do. And that got us thinking whether or not then this would be something that the virus might do. why am I suddenly not advancing? Something that the virus might do naturally. So what we did then is we took HIV sequences in GeneBank and we used them to query the human genome. And we could easily find sequences that matched our expectations. So what I'm showing you here is, for example, a circulating recombinant form where we're having 100% homology to one strain, and then there's a break, and then there's homology to a second strain. And at that junction, then you can find this very close match to the human genome. But then the question becomes, am I seeing things I want to see? My junior high school algebra teacher would have been disappointed in me if I'm trying to do that, because it's sort of it was kind of a mixed blessing. Those sequences I showed you are microsatellite sequences. So on the one hand, I could understand the virus might use them because it would frequently encounter them, but I also couldn't really assign the parentage of those sequences to any one allele in the human genome. So we looked a little further and we found this paper in the literature where there was this so-called foreign insertion in an HIV strain. And what happened is that this was a story about a young child who had been infected perinatally, and he had a pretty normal reverse transcriptase gene for forever until he was treated with AZT. And then all in one step, he got this big insertion, which was foreign to these guys, which then conferred resistance to AZT, very interesting mechanism. But important to us who think about horizontal transfer and so forth, was that this insertion had an unusual GC content for an HIV. So I happened to meet this man at a meeting, and I had told him about my model, and he encouraged me to go ahead. So I pointed out to him that yes, we search, and at least at the time, this sequence insertion in this child's virus, its very closest match was to human genome, and it really didn't match anything in any kind of viral genome. So we came up with this model where, again, this virus must have inserted in chromosome 17, read through, and then what I'm showing you here is sort of this wrapped around tail of the read-through RNA. And then we just have to, again, postulate that the microhomology, not that small really, 4 on this side, 6 on this side, would patch this in. And then there would have to be a few additional mutations that would arise in order to make the sequence that was published in the paper. But it was kind of fun to do the molecular archaeology with that investigator from Japan. And within the child, there was, of course, a swarm of viruses, and the changes that they had were all quite consistent with then this original founder strain and entering up into this viral diaspora that had given this child that property, his virus, the property of AZT resistance. And so I think it's kind of a cool thing, but that would be the end of this story, except for 2 more things that I wanted to tell you. And the second one is really remarkable, which is 7 years after I published that model, then these people in Belgium reported another virus that had an insertion very similar to the one that had been seen in Japan. So it was 1 codon shorter and a little bit different, and the backbone was clearly different. Clearly, this Belgian strain, the HIV, was totally unrelated to the Japanese child, but remarkably, it had inserted sequences from the same host locus, and then made the case that this was actually a transduced sequence, that much stronger. So there's lots of cases where we see these little insertions in HIV, and they're usually so short that it's pretty impossible to say that's really where they came from. But it's what the enzyme does. The enzyme doesn't get to the end of the template and just improvise. What it does, what all our non-homologous recombination junctions showed, is that it does do this kind of splinted recombination. And the other thing then that's kind of fun to think about is that there are other viruses. So this is an old slide showing us coronaviruses, for example, that their polymerases are really highly accustomed to do mechanistically basically the same reaction, that they jump between short patches of homology. And this virus also recombines. And so I think that when you hear people say things like, there's a coronavirus, we won't name any names, And it looks sort of like one virus and it sort of looks like another virus, but it doesn't look exactly like those. Are we missing a parent of that virus, or was it made in a lab? We don't have to postulate such nefarious origins for these little inserted sequences. There are always other ways. So there are always other ways. And I don't know exactly why I gave you the little points in my life that I did. It was the last couple of days throwing away a lot of things that had made it too long. And the main thing that I regret is that life is not just a series of moments, of course, but it's people. And I couldn't talk about many of the people who were supremely important in my life and who make this meeting really important to me. And especially, perhaps I should mention, my current grad students who are fabulous and who have stories that are completely unrelated to anything I talked about today, but who you will hopefully hear from in the future. But I did want to come back to the meeting for just a moment at the end of my talk and point out to you again about how we have such a wonderful community. And so a lot has changed in the last 30 years, and we are all at the remote retrovirus meeting today. But I'm really proud of our field, and that we've seen it again today and these last couple of days, that we are really supportive of the junior people in our field. And I wanted all the junior people in the field to see this slide, to see all these leaders in our field who came together and endowed a prize, a career development prize, to point out that this meeting is about the junior people, and what we care about is the advancement of people in this career, that you keep us feeling lively and enthusiastic and wanting to be here. And I wouldn't be doing my job if I didn't point out to you guys that you can still donate if you would like to have your name up here. I would be happy to tell you about this, and the information is also on our webpage. And I did want to talk, we've heard a lot about how this is like the 46th annual meeting and such. And this meeting has a history, and we talk a lot about the old people and the old things, and I did that today. What can I say? But this meeting does evolve. So for example, although I guess viruses evolve faster, but meetings evolve too. These are the first 10 abstract books that I got. Up to the year 2000. And way back then in Y2K, and I had this stack, I noticed something funny about them, which was that out of the 20 organizers that were represented in this 10 years, only one of them was a woman. So I mentioned that fact to the organizers that year, and I don't know if that influenced them in any way, but I guess what I would like to point out to you, and we've all seen, is that we have a lot more gender equity in our leadership ever since. And although the task of making this meeting leadership better represent its participants remains a work in progress, I think that an old timer like me nothing makes me happier than seeing session participants from all around the world, old participants, young participants at all different stages of their careers, duking it out here over new discoveries. So I think that when we think about the pandemic, and if we've learned anything from it at all, it's that the health of the public requires not only science, but the buy-in of individual people. And I think that this meeting allows us to interact with one another and work things through together. And it's worked well remotely, nonetheless. I hope you'll all stay safe and we can see each other next year in grace. Thank you.
Thank you so much, Alice. That was fantastic. I guess I gave some foreshadowing for your mentorship and teaching interests, so that was great to hear about. I don't know if anybody has any specific questions for Alice. Feel free to put them in the Q&A. I guess in looking at your, these inserted sequences that you're seeing from in these patient isolates from human genome, is there a reason why? I mean, how frequently do they occur? If it's relatively infrequent, I could see that they would just get selected out. But if there's some sort of advantage, you would maybe see them stick around.
Right. And I think that's the thing that was probably demonstrated more by other people than myself, is that non-homologous recombination is less than 1% as frequent as homologous recombination. So it doesn't happen all that frequently. But still, we talk about how many different viruses we think are arising, but it's not driven by anything selective. It's just by nucleotide homology. So again, most of the time, it's probably junk and just lost. It's inconsequential. But I guess I still am blown away by the fact that this one sequence ended up twice in a virus, and it kind of, it's interesting. And as I said, I can sort of look and see, a lot of times when you see these insertions, they're from discontinuous part of the viral genome, which makes sense. It's exactly the same mechanism that I described. It's just using a patch from within the viral genome. But yeah, these things happen pretty often, and they just are either useful in the starting point for more evolution, or they're lost.
So a lot of people are saying great talk, very inspiring. David McDonald is very happy that you promoted his meeting. So good job.
You should go. It's really a great meeting, although this meeting has been fabulous. Sandra,
yes.
You're doing a great job.
Well, thank you. Stuart is also involved in that, of course.
Yes, of course.
You didn't talk, like, I know that you have a lot of students from Africa. I think maybe Ghana you have a collaboration there. I don't know if you're still involved in that and how that works with the University of Michigan.
Yes. So it's kind of a funny story, actually. So that picture that I showed you, and I don't know, maybe Eddie will not like me to tell this quick story of Eddie, but I was already interested in students from African countries, and probably again it came from when I was on sabbatical. I said, why don't I branch into HIV? And I went to a meeting, and this fellow from Cameroon came up and talked to me, and he seemed really brilliant. And I said, where'd you get your PhD? And he said he didn't have a PhD. So I just, at the drop of a hat, decided to get him a PhD. So he came and joined my lab, and he's now doing COVID surveillance in Cameroon, which is his country he's from, but he was working in Zambia for a while and so forth. But anyways, it was such a pleasure to help him get a PhD, and he's now a leader in infectious disease on the continent and so forth. So with Eddie, so we've had a number of students over the years, but Eddie, actually I was not encouraging him to join my lab, but he came to my lab as a volunteer, as an undergrad from a small, basically sort of like a community college in rural Ghana. And he just did great. And so then I wanted him to come back, but we couldn't put him in our PhD program yet. So, you've met, of course, Eddie. And so in fact, then I got this brilliant idea, because this is what I'd done with the fellow from Cameroon, is I sort of ad hoc arranged for him to be in a master's program in his home country, because then the tuition was cheaper. But when I had done that before, they just sort of let me register my previous student, and it was kind of a paper degree, but the guys in Ghana wouldn't let that happen. So we had to actually do the training thing. And ever since then, we've been kind of on this collaboration. In fact, actually, I have upstairs in my house right now, even as we speak, a new student who just came from Ghana who's going to do his PhD in my collaborator's lab. So yeah.
Wow, that's amazing. Monica Roth is asking, or she mentions that you faced a period where you thought about leaving science. I didn't know that, but she does. And she wants to know what gave you the strength to return.
What gave me the strength to return? Well, it's interesting. And again, I guess I'll tell this little story, which I've been having fun telling it. And I think Monica actually knows this. The reason I thought I was not going to be in science is that when I was an undergrad, one evening, my mentor called me into his office and said that I didn't belong in science and I should get out of his office right away, out of his lab right away. So I did. And then I was just sort of wandering around. Actually, I was in Asia. And then the man wrote to me and said, would I forgive him? It was something about my eyes. Wouldn't I come back? He couldn't live without me. So that was how I had fallen out of science. Was one of those Me Too type moment things that you don't want to have happen to you. And I think that somebody else was pointing out that words do matter, and you never know which of your undergrads is going to do well. And as an old lady now, I'm very resentful of the years I lost because of that man. But those things happen. So as I said, actually, it was, this more sort of life is random sometimes. And I came back to the United States just for a family visit. And then my boyfriend in Japan, who was an illegal alien from Hong Kong, got arrested. And I met Herb Heinecker in an elevator, and the rest is history.
Wow. That is a very interesting story.
It's too much personal, Karen.
So Chris Aiken says, Alice, I've always enjoyed your perspective on science and our field. Thanks for sharing more of it today. What is your best advice to science PhD students today?
To science PhD students today, I think that you need to know that your skills and training are so valued, okay? And one thing I would say to you right off the bat, and this is a cheap trick, which I was afraid Zandria was going to say because I said it to her. All of you mentors, whenever you meet a brilliant young person at Cold Spring Harbor, tell them you want to postdoc in your lab, because if nothing else, they'll just think that you're very prescient and you recognize talent when you see it. And everyone can use a little boost. But I think science is tough. And so those people have to realize that the feeling about it being tough is something that we all have. But the skills, the kind of critical thinking that you're developing are invaluable to so many different fields. And yeah, I don't know. I think also do talk to, like next time, if we do get together at Cold Spring Harbor and you see me just sitting there having breakfast, come over and talk to me. And I think we should have more of our faculty talking to trainees over breakfast and so forth, because yeah, wasn't the best answer, but we'll go ahead, right?
Yeah, I think that's something that we all miss. I mean, we miss Cold Spring Harbor, interacting with people, but one of the fun things is to come in at breakfast and sit next to somebody you don't know, right? And engage in a conversation that you wouldn't normally have with somebody. So yeah. Well, I'm not seeing any more questions. You've got a lot of kudos. Everybody really enjoyed your talk today. Thank you so much, Alice.